Bismillahirrahmanirrahim. Welcome back to the second part of characterization of nanomaterials and nanoparticles. In today's lecture, we are going to discuss the electron microscope. As we know that light microscope cannot resolve structures closer than 200 nanometers. That's why electron microscope is being used to have a greater resolution power and magnification powers, and it magnifies object more than 10,100,000 times and 10,000 times. Detailed views of bacteria, viruses, internal cellular structures, molecules, and larger atoms are possible using electron microscope. Energy source is a beam of electrons. An image is created and viewed on monitors. There are two types of electron microscope: transmission electron microscopy and scanning electron microscopy. First, we discuss scanning electron microscopy. The basic principle is that a beam of electron is generated by a suitable source, typically a tungsten filament or a field emission gun. The electron beam is accelerated through a high voltage and passed through a system of apertures and electromagnetic lenses to produce a thin beam of electrons. Then that beam scans the surface of the specimen, means sample. Electrons are emitted from the specimen sample by action of scanning beam and collected by suitably positioned detectors. This is an instrumentation for scanning electron microscopy. Now we will discuss the working of SEM using the above instrumentation parts. Electron gun. An electron beam is emitted from an electron gun fitted with a tungsten filament cathode. Anode. Electrons accelerated towards the anode, then the condenser. After the beam passes through the anode, it is influenced by two condenser lenses that focus the beam onto the object. Objective lens focuses the beam onto the object. It does not image the specimen. The scan coils deflect the beam in the XY axis so that it scans over a rectangular area vertically and horizontally of the sample surface. Sample. Focused by electron beam. Backscattered electron detector BSE are beam electrons that are reflected from the sample. Since heavy elements, high atomic numbers, backscatter electrons more strongly than light elements, low atomic number, and thus appear brighter in the image. Backscatter electron detectors are used to detect contrast between areas with different chemical compositions. Imaging. It converts the electron image into human eye perspective form. Image is digital captured for display on a computer monitor. Note: sample and electron beam should be placed in vacuum. Specimen should be very thin and dehydrated. Advantages. It gives us powerful magnification, high quality resolution, provide information on sample surface and its composition. Disadvantages. Scanning electron microscopy is large and very expensive instrument. Its sample preparation is laborious. Operation and analysis required. Special trainings. Requires special